So welcome to the course Data Science in Cell Imaging. We have here guests from, not from BGU, right?
Right.
Okay. So I think I'll start. I have pretty quickly in the presentation. I have questions and answers, and then I also want to hear a little bit about the students who are taking this course, not from my department, to hear a little bit about your background and try to see how we can fit everything together. So I'm starting sharing my screen. Okay, so welcome to the course. This is the second round of this course. Last year was the first round. This year, I decided after, I think, mostly a successful run of the first run to open it to the public. So in principle, anyone who wants to, so I advertised it and people asked for the link, and I'm open to anyone who wants to join and hear. I have motivation there because I want people from diverse backgrounds. The students that this course is designed for is computational students with no previous background in biology at all. I would like to have people with a little broader perspective in terms of the biology and other domains. And I would like to have these people also speak and educate me as well. I mean, I'm not an expert in every aspect of, in most aspects of biology, to be frank. And this is definitely part of my motivation. Also, this course, I think, is quite unique in terms of the materials that it covers. There is no textbook, and most of the material is based on papers from recent years. So please, so I want to open it to everyone that can learn from that. Okay. So these are my contact details. I'm back at the office now. So if you're in the third floor of our building, you are invited to come and say hello. And yeah, very, very happy about that. And okay, so let's start. So the idea of the course is to review state of the art in processing of visual imaging in microscopy. And I think that the main difference to other bioimage analysis courses is the idea that we're interested in finding the pattern. I mean, we're not interested in the standard bioimage analysis stuff. So doing better segmentation, better tracking, a better, I don't know, alignment, registration. But we're interested in answering a specific any questions to me already, or is it specific biological questions? So the motivation is actually the biology, and not necessarily having a fancier computational algorithm or something that will do better segmentation, but how we can use the output of these algorithms, which are to extract new biological knowledge. I think one of the key aspects of this course is that it's very diverse. So the biological questions are very diverse. I mean, the one common theme is that it's about bioimaging, right? So microscopy imaging. And in terms of computation also, you're going to see a broad and diverse set of techniques that we are going to use from very different domains and fields. It also implies that the course is going to be, we're not going to go deep. We're not going to go deep into deep learning, into machine learning, into time series analysis, into a specific biological domain. going to be very broad, and you need to take it into consideration. Okay, so I'm continuing. Why am I speaking English? So once because I want to practice my English, but more seriously, I think I want to open this course to the world, and I want people to be able to join us and yeah, maybe share the share the information here with as many people as possible. Also, I think that the language for science and it should be English, and this is the way that science is communicated. I want to say that I'm expecting here people to participate and ask questions. Don't worry about English. You can see even I'm the lecturer and my English is not perfect, far from that. And the idea is this is a supportive environment. Use it as an opportunity to train your English. And if some word you cannot find it, you cannot just say something similar, or just ask in Hebrew and we'll translate. The terminology is also kind of, because it's very interdisciplinary, the terminology is also going to be, very diverse. So I'm feeling completely comfortable people stopping me and asking me about a specific word or term, or to explain things. I mean, it's completely fine, okay? But it's going to be in English. Questions that usually people ask me: do I need any background in biology? The answer is no. I assume no prior knowledge in biology. I do expect some interest in the domain. I mean, if you are coming to this course from other, I don't know, agenda, then it's not the right place. I mean, it is, I think, a kind of a niche, right? It's a very niche topic, or a very specific topic, data science in cell imaging. So I'm expecting people who do want to learn about this domain, and not just to have a course to mark with their grade sheet. And if this is the reason that you come here, I mean, it's a mistake. You're going to suffer from that, and it's not. I mean, it's really not a good idea, I think. And I'll explain a little bit more about that when I'll tell what I'm expecting from this course. So last year we were actually only 14 people in this course, and this year I think it's full house. I mean, I don't know how many people registered to it, but it's about, I don't know, above 40 officially are listed to this course, maybe even 50, I don't know. So I mean, and it was fantastic when we were 14, because each and every one of us were interested, and it was like the discussion was lively. All the cameras were open. I'm going to talk about that as well. I mean, if you are here, please open your camera. And I'm expecting it to be even better this year. Computational background is required. I mean, this is a course to take computational students with computational background, and bring them and show them a little bit about the fascinating and the exciting world of cell biology. So yes, I am expecting students to have some computational background. I am going to give a very brief description and intuition of every techniques that I'm going to present here. I mean, realistically, I mean, I'm you don't need to have a very broad idea about any specific computational aspect, because I'm going to give it to you, but you do need to be able to know the general terms and to think in a quantitative way. So yeah. Questions? Okay. A few students asked me if it's a good fit for an undergraduate students, for mostly from the department. So last year we had the two third year, very good undergraduate students. And I asked them that explicitly at the end of the course. They say that they enjoyed the course, but it was very challenging to them. So if there are here undergraduate, please keep in mind that it might, I mean, it's your call, right? I mean, if this is really what you want to do, then I'm not going to say no. I'm going to approve anyone who wants to join the course. But please consider that it's going to be challenging, especially the project, which is, I'll talk about it in a few minutes, which is, I think, it's pretty, you need to be pretty mature to be able to get something reasonable out of your hands. This is not a deep learning course, okay? So if you are here to learn about deep learning or any other specific technique, please, there are very interesting and comprehensive courses about machine learning, about deep learning, time series analysis, network algorithms, data science in network in the department. I mean, really, they are fantastic, and please, this is probably the right place if this is what you are looking for. And the course is not going to be, I'm telling you all of this because it's kind of aligning expectations, because last year I had here, it wasn't perfect. I didn't realize that at the beginning, and some people were disappointed by that at some point. So very few, but there is, I want to make sure that if you are here, you know what you are going to get. So the course is not a hands-on course. We're going to discuss ideas, and your tasks are going to be basically presenting a summary of a paper, and then the only hands-on assignment is going to be a final project, which is a research project. This is going to be hands-on, but beyond that, everything else is going to be, we're not going to program in this course. But the project is going to be involve, of course, coding. Attendance, it's not obligatory. You can decide to come or not to come. The lectures are recording. I think I recorded now, right? I mean, can you see that? Yeah, I don't see it for some reason, but it should be.
Recording. Yeah.
It is
okay.
recording.
Okay, so yeah, it should be recording, and you can come or you can decide not to come. I do expect the lessons to have discussions. So I'll be happy if you come. If you come, please open your cameras. I mean, it's not, if not, then just watch it offline. Why be here physically? I mean, I'm here to see faces. I'm here to communicate with you. I'm not here to just because otherwise I can just record my lectures and bring them up, and so I really ask you to do that. The course is challenging. I mean, we're going to have the terminology is difficult. The way of thinking is very different from what we're used to. The final project is pretty. If you want to get something, to get something meaningful, it's pretty challenging to do a research project in a very difficult domain as a task for a course. The articles that you are going to present are very long compared to what you are long and complex in terms of the amount of data compared to what you are used to in the computational domain. You're going to spend a lot of time on reading them and understanding them and finding, squeezing what is most important out of them. So I think it's above average in terms of time that you are going to invest in that. And the grades, I mean, last year the average was 90. The class was exceptional, so I think it's it's an upper bound. It's not going to be better than that. One student said just couldn't out of 14, right? Couldn't finish the final project. 3 students got grades around 70, I think. So I mean, it's not necessarily a course with very high grades. So please stay here. Well, you get the point, right? I mean, please stay here only if you really want to be here. COVID. I'm starting with remote teaching. I hope that after, keep your camera on. You see that it's a theme that I really want to see your faces. After Passover, I hope that will be back at the university teaching. I'm going to take this class as a hybrid class, which means that I'm going to be in a class with students. Whoever wants to join can come. I'm not sure if they'll require a green passport or not, but anyway, I mean, probably there'll be very clear restrictions and definitions who can join and who cannot, and it means that that there is it's a classroom with a camera that is following the teacher and good sound. I didn't do it before, but I heard that it works okay. I assume that for people who watch it remotely, it's not as great as as just direct Zoom because because the teaching is in class. So you should also take that into consideration when deciding on whether to stay or not. Okay, so today I'm going to give a very broad introduction. I'm going to talk about the administrative details of the course and give an overview of the schedule of the course with some highlights. Actually, I brought some highlights that most probably I'm not going to show in the course because there are, but we'll see. I mean, but it's just to show a few examples of things that are relevant to the course. And yeah, before we start, so how many of you are not from the department?
I'm not from the department, but like.
You are from biology?
Yeah, I'm a master's student in biology and molecular biology. So.
Okay. Who else?
Me, hi. I'm from Tel Aviv University.
Yeah, but you are a computer scientist by training.
Right.
Anyone else?
Hi, I'm a final year undergrad student who came here as research exchange. I'm originally a computer science student, but I do data science for the Department of Chemistry at BGU. Okay,
welcome. Okay, so people with a little more biological background, please correct me if I'm saying something wrong. Okay, please feel also comfortable to stop and ask questions about the computational aspect. But I really want, I'm not going to. I don't know everything here. A lot of things I do not know perfectly. So please, Noga, this is your job, right? Good. Okay. Alina, you. Okay, so let's start. So cells are the fundamental unit of structure and function in every living system, right? Our body is built out of cells, plants, anything in the world, basically. This is the atomic unit of function within a multicellular organism. And cells are built out of molecules, and the molecules are defining the structure of the cells and the function of the cells. So in a cell there are thousands of molecules that are building how the cells look like, its morphology, and also define and also in order to operate properly, you need every molecule to come into work with a very synchronized manner and in and coordinated manner with the rest of the system in order to get the cell to do what it is supposed to do. Oh, you can hear me? No, you cannot hear my sound, right? How do I do it?
What is the meaning of cells tick?
Cells what?
Tick.
Tick. It's just slang for actually do what they're supposed to do. I'm just looking to how to get my sound.
You need to share your sound.
Yeah, how do I do that?
From the share icon.
The share, screen sharing. No. Come on. Usually I find it. I don't know why I select the speaker. Okay, I'm just going to run it and without sound and then describe it. So this is a very famous movie from the 50s of a neutrophil, which is a white blood cell, which is actually chasing a bacteria. So the neutrophil is what you see. The neutrophil is what you see here, right? These are the red blood cells, all of these guys which are not doing much, and this is the bacteria. And when I'll continue playing this movie, you'll see the bacteria is chasing, the neutrophil is chasing the bacteria, and the bacteria is running away from it. And also notice that there is another bacteria over here, for example, that is not doing much. So it's not, you know, it's something that occurs very specifically. Okay, so what you see here, I mean, you can see that the neutrophil is chasing the bacteria. It's very clear that what's going on here is not random motion, right? I mean, the bacteria, the neutrophil is sensing the bacteria, is making decisions based on what the bacteria is doing, and the bacteria is doing the same thing, right? I mean, it's sensing that the neutrophil is chasing it. It's running away. It's trying to make turns and flips in order to escape from it. And eventually, in this case, the happy ending is that the neutrophil is winning and taking, you know, killing the bacteria. Anyway, in order to do all of this, these cells must have a very complex machinery that allows decision making. So it allows the neutrophil to sense the bacteria and vice versa, to process information regarding the sensing, to change its internal state, and then to make decisions in terms of changing its internal machinery, which are the molecules, which I showed in the previous slide, in order to move in the correct direction, make turns, and eventually catch the bacteria. And the bacteria does the same thing. What is enabling all of this intelligent system or complex intelligent system within the cells is the molecules, the proteins within the cells that are working together, processing information and making this type of decisions. And the only way to actually understand what's going on, to follow these dynamical processes, is to use microscopy, to look at live images of cells doing stuff. Because if you take snapshots, you cannot now, from this snapshot, you cannot really understand that now the bacteria is going to take a right turn and the neutrophil is going to take also a right turn, and which molecules are coming to action in what time and in what context in order to get the things to work the way that they are supposed to do. So in order to understand dynamic processes, one of the best ways is probably microscopy. And this is not new. I mean, for hundreds of years now, biologists are looking under the microscope, seeing what they can see, describing the behavior and the appearance of the cells. And biology have worked this way for hundreds of years now. And most of the modern biology is based on this mode of action. The person, one of the persons who starts that is Robert Hooke, who's the father of microscopy, and he is also the person who termed the term cell. And basically, this type of images, which are a dead leaf, right? A dead leaf allowed him to see these honeycomb structure and this I don't know, this very structured element, which he called cells because they look like a cell in this honeycomb. And these are actually the cells that we're talking about. These are specifically dead cells. And he developed the first microscope, which you can see here is how long ago? Over 400 years ago. And this is the way that things work until now. I mean, we're looking under the microscope and seeing a phenotype and describing it. I want to go through two major breakthroughs in microscopy that are a little more recent. So one of them is the discovery of the green fluorescent protein of GFP. So you saw the neutrophil chasing the bacteria, and we talked about thousands of proteins that are playing a role together and coordinating the cell's behavior in order to make decisions. But can we see the molecules? Can we see each molecule? Where does it, each molecule, or in general, the molecular structures, how they come into action, and to see them visually within the microscope? So with live imaging that I showed you before, I mean, no, we do white light, we see the cell, right? I mean, we don't see the internal structure of it. But these three people who received the Nobel Prize in 2008 in chemistry, they discovered the green fluorescent protein, which is basically the ability to use a protein that comes from medusa. How do you say medusa in jellyfish? Yeah, jellyfish. comes from a jellyfish and emits under a specific wavelength, and label with it a molecule of our interest. And once we do that, we can start looking at the molecules of our interest. So what you see here, okay, on the left you see the jellyfish, but on the right you can see the cell, here labeled in three colors. Blue is some molecules that is labeled the nucleus of the cell, and green is, I don't know, what is it, maybe actin or microtubules, and red is, what is it, a membrane micro, I don't know. Anyway, and this allows us to look at the cell and start to see the internal component of a cell. Now there are thousands molecules within a cell, and we can see only a few of them in each living cell, and I'll talk about it a little bit later, why and how. When we talk about scales, we can talk about spatial scales, and we can talk about temporal scales. And micron is the 1 millionth of a meter, right? So when we talk about cells, we talk about scales that are tens of microns. And we can start looking at the internal components of the cells, looking at the micron scale. And beyond that, basically, is the limit of light imaging. So this is what we can get with light microscopy. And if we want to get better resolution, we need to go to other techniques, which I'll briefly describe. And also, we have Natalie Elia from Life Sciences coming next week as a guest lecture to give us a broad introduction of cell biology and microscopy. And she'll also talk about these ideas. So we have a resolution limit, which I'm not going to go into detail. Again, I'm going to leave it for Natalie. But basically, think about it. If we have a very small point, one molecule that we want to image, still because of optics limitation, we're going to see a halo around it, and we're going to see, and basically the resolution that we get is 250 nanometers. So 1/4 of a micron, and yeah, and this is what we are going to get. And if we have a few of these, so if we have 3 point sources, we can actually, and this is how we're going to see them under the microscope. Oh, this is my marker. Okay, so if this is what we get. If we have 3 molecules that we are interested in, when we look under the microscope, because of the resolution limit, we're going to see these 3 halos, and then we can localize that and understand that these are the points and make a distinction. But once we have too many of these, everything is starting to blur, and it's going to be really hard to distinguish between the different point sources. So the next innovations that I want to highlight is super resolution. And super resolution is the idea that we'd like to take an image that you see here on the right and turn it into a more detailed image where we can see the details. So for example, in this image, again, I took a slide from Natalie. You're going to see everything is very blur, and super resolution is improving the details here and being able to see the specifics. So now, if at the beginning we cannot read the signs here, after we improve the resolution, we can do that because we get rid of the resolution limit and improve it. This idea is made possible in microscopy by Eric Betzig, William Warner, and Stefan Hell, who developed the super resolution microscopy. The idea there is since we cannot resolve these structures if we have two dense sets of point sources, what we can do is activate stochastically every time a subset of these points. So we are not getting the whole picture in one time point, but we're going to each time to induce stochastically part of the points. And then we're going to get a less dense set of points, and we can resolve each of these images. And then if we do it multiple times, we can eventually get the whole picture, as you can see in this movie. So on the left here, you can see the blinking dots. Each of these dots can be localized to a very specific dot. And then on the right, you can see the image resolving over time and becoming like a very clear structure, while on the middle, you can see the initial resolution of the original image. So this is a very simple idea if you think about it. I mean, it's basically a very trivial idea, both computationally and the advances that allow to make this technique is actually in the stochastic lighting illumination and turning on and off of the molecules. But it's still, I mean, it allowed now to improve the resolution, and this is so important that it got a Nobel Prize to the people who invented that. And an anecdote about that. So Eric Betzig in his talks is usually showing this super resolution microscope that is built in his. Actually, I think it's not his. It's in his. I think it was the other. It was in. I think it was in Stefan Hell's living room. They were both unemployed. They had this idea, and they built this microscope, and and then they found the job. Kind of. I mean, this is how he tells it. It's not completely true, probably, but you can read about it here below. So there are a few ways to do that, to resolve the structure of super resolution. And there's a few techniques about it. Natalie, again, is going to talk about it a little more next week. But I really like this movie that illustrates this process. So you can see if we start from the beginning, you can see that each time frame, we get an image of only a few of these dots that are lighting up. We can do a localization. And by accumulating this localization over time, we can start getting the structure of what we are looking at. Is it clear? Okay. Any questions? Okay. Okay, so light microscopy, this is the limit. 250.
I have a little question.
Yeah.
For the super pixel, we take an action just with the classic methods or some deep learning methods for make it happen?
Okay, so the idea here is it's not, there is no, I mean, the computational aspects are pretty simple. What all that is done here computationally is localizing these point sources. And yeah, now you have techniques that do that using deep learning, but in principle, you can do it with very traditional image processing methods, okay? So, I mean, this is, yeah, so there is no, but today, and one of the examples that I'm going to show you in the lessons that I'm going to give about applications of deep learning in microscopy are going to involve how we can do that with much less frames. So here you need to take many frames, and each time stochastically some of these point sources are lighting up, and this allows us to eventually find the structure. Later on, I'm going to show you how deep learning was used in order to do that much faster. Yeah. So you don't need to take thousands, I don't know, or the hundreds or a lot of images to actually get a structure, but you can get it much faster, and this allows you to do things that you cannot do with this low temporal resolution. Okay?
Okay, thanks.
So think about it. I mean, there are two things here. One is the ability to stochastically activate, right, to light a molecule, right? This is one. And second, which is quite simple computationally, is doing the localization itself in the image. Okay, so light microscopy, these are the limits, and with super resolution, we can get a much higher resolution. But if we want to go even further, we need to use another technique, which is called the electron microscopy, which I might talk a little bit later in the course. I'm not even sure if we'll get to that. Anyway, these are the spatial scales. And now let's talk a little bit about time, about information in time. So in microscopy, we have space, our information in space, and information in time. So the second thing is temporal information. And here you get 3 snapshots in time. And the question is, I mean, when we look just at one snapshot, the question is, what can we learn about what's going on here? I mean, what direction is the ball moving? What direction are the players running? Who is running fastest? Who is who is making the accurate passes? From snapshot, we cannot make this distinction. We cannot understand what's going on. So in order to understand dynamic processes at the time that they happen, there is no other way but microscopy, live imaging of whatever we're interested in to understand what's going on. So now when we see the video, we can see very clearly, exactly all of these questions. We can start answering them. I'm going to, this is not completely true, because some dynamical processes, we can take snapshots at different times of different cells and learn quite a lot actually about the dynamic process. For example, developmental processes. I mean, we start with, we start with a small bunch of cells which are growing and becoming whatever, human being or a model, I don't know, a zebrafish or Drosophila. And there is a lot of good science that's going on by stopping and taking snapshot images. But if we want to actually focus on what a cell is doing and how cells are making decisions and taking action in time, we do need to have the live imaging. Okay? Like in a soccer game, we can still understand the end result if we look at the game and see just the times where, I don't know, see the scoreboard, right? I mean, we'll still be able to understand how the game went and or seeing just the statistics. We will be able to understand which player was good at what, but of a basketball game, for example. By the way, I'm going today to a basketball game of Hapoel Be'er Sheva. Pretty exciting after a year of not being able to do anything like that. But yeah, so these things we actually need to see what's going on live, because we cannot extract all the information from snapshots. So I talked about seeing is believing, but in the last 2 decades, I think we moved another step ahead. And this is not enough, because our eyes are misleading, and we're biased to nice images. So if we see something interesting in the microscopy images and we want to say, oh, this is what's going on, we might be ignoring a lot of other things that are going on, which are not which are describing better the process. And also we want to be sure that what we are saying is general and true and not something very anecdotal that we see. So the idea is that it's not enough to look at the pretty pictures. We need to put numbers in order to prove that our hypotheses are actually true. So we see something and then we want to quantify that, to put numbers on it, to test the hypothesis, whether it's true or not. And this is especially true. So at the beginning, or even now, a lot of poor graduate students in the more biological departments are counting molecular events or counting cells or check, or doing very manual work in trying to quantify, to put numbers on what they see in microscopy images. This is all, I mean, it's cheap labor, it's all good and fun, but I mean, for the students, it's pretty terrible. I mean, I don't understand how some of them survive annotating thousands and more of events. And practically, with the new era of big data generation, this is not practical anymore. We cannot do it we cannot work this way anymore. So here I'm showing you 3 imaging techniques that generate a lot of data. I mean, what you can see on the right is the production rate. I mean, it's true that this production rate are upper limit. So if you use the machine to its full power, and usually they are not, but still you can see, I mean, the numbers are huge, and there is no way anymore. I mean, even just storing this type of data and doing a very, I mean, even counting something that is very trivial and easy, it's going to be very hard when we talk about these amounts of data, and it's just not practical anymore. So we need quantitative tools to do that automatically. And there are a lot of free and open and also proprietary tools that allow us to do that. So what you see here, Fiji and the Cell Profiler and IC and Elastic, all of these are fantastic tools that enable us to take an image and to do the standard processing steps, segmenting cells, detecting molecular events, etc. And that's great. I mean, this transformed cell biology, I think. And now I think there is still manual annotation going on, but a lot of the manual annotation now is gone, and these tools can be used in order to resolve that. What does it give us in terms of a field? So the ability to automate the processing gives us 2 things. One is automation, so avoiding the need to do manual work. And the second is completeness. What is completeness? The ability to appreciate the full distribution, the full variability of the data. So if before we were focusing on a specific, specific cell that we were biased by and counting something specific within it, thanks to automation, now we can look at the whole heterogeneity, the whole diversity of the population, and we can appreciate the true complexity of the system. It's also very important. Completeness is also very important when we want to focus on rare events. So think about the cancer. We want to look at the one cell that is prone to, I don't know, to create the metastasis eventually, and this gives us the ability by characterizing the whole population. Most of what we're going to talk in this course is not about these bioimage analysis tools and quantification, but about the third leg that is missing down here, that is going to join soon. And the idea is that that sometimes we need to switch the order here, because our eyes cannot teach us on the complexity of the process. It's just too complex. There are too many things that are going around at the same time. Our eyes are not trained to look at this type of events. And even if we are staring at this type of images, it's just too, the patterns that emerge them are just too complex. We cannot appreciate that visually. And then sometimes what we need to do is to switch the order. First, extract quantitative information about what's going on in the microscopy images. And once we do that and we find interesting patterns, we always need to go back to the biology and try to understand what we learned, what is the biology that we've learned. I think this is very different from a lot of what is done in the department in terms of applied computer science, where what we care about is the ability to be better than our competition, right? So the state of the art is here and we are here, okay, great. I mean, this algorithm is cool and it improves the state of the art. In biology, this is not enough because we want to understand why. We want to understand what is different exactly, and not just show that we're a little bit why we're better, and not just that we're better at something, right? And what is different here that we want to characterize. So, and this is a very challenging task because we need eventually to really go back to the biology and understand what's going on there. In order to do this switch, in order to switch these things, we need a very deep domain knowledge. So we are a kind of applied computer science or a data science department. And probably you heard about also in other courses that, you know, we have a very broad and impressive toolbox that can do a lot of things, can solve a lot of problems. But if we're a true data scientist, we cannot just come to a new domain and say, hey, I have this fantastic toolbox and I'm just going to throw it and use it and solve all the world problems. You actually need to understand the domain that you're working at. And I think this is especially true in such a complex domain as cell biology. If we know the domain knowledge, we can understand the experiment. So if we see something, we can go back to the experiment and understand what went wrong, or why did it work, or why it did not work. We can, I mean, I'm not an experimentalist, but I can sometimes I go to my collaborators and ask them to do the experiment a little bit differently, because I know a little bit on how to do these experiments and I know what can be changed. And also, once we do an experiment, we need to know what to look for, what is interesting to look for, what question to ask, what pattern to look for, where to look for it in the image, and what tools are available out there to extract this information. And then eventually we need to go back to the biology and interpret this information in order to come to a conclusion. So the third leg here of the contribution of computer vision or bioimage analysis to the field of cell biology is the ability to identify patterns that are hidden within a very complex data. And for that, I mean, I showed you earlier techniques that are generating a lot of data. This is not necessarily what I mean, even in one image, or even in you know, one movie, one time-lapse movie, which is not huge in terms of you know, gigabytes of information necessarily, there is still a lot of things that are going on and could be also big data and complex data in terms of the ability to interpret it and find the patterns within it. Okay, I think it's a good time to go out for a 10 minutes break. So we'll be back at 3:17. I'll stay here if you want to discuss, and we can turn into Hebrew, whoever wants, about if you have questions about technicalities. I'm going to talk about the administrative issues, but if you want to talk to me about anything else, then I'm around.
Can you ask about the project?
Excuse me?
Can I ask about the project?
Sure.
Is it in pairs or individual?
Yeah, whatever you want, whatever you prefer. So it could be, I would say 1, 2, or 3. Okay. I mean, 3 is preferable, not 3.
And can you a bit elaborate about what kind of project it is, what the direction, what we're supposed to investigate in there?
Yeah, but I'll do it in the last part of the lecture today.
Okay, great. Thanks.
אם מישהו מתבייש יכול לשאול אותי גם בעברית. טוב, אז גם אני נעלם, נתראה עוד 9 דקות. Hey, Emily. We're in a break now, so we start in 4 minutes.
Oh, okay. Thank you.
And I think most of what I've gone through, I think you know. So
Okay, thanks.
okay. Let's continue. Can you open your cameras? Please, whoever can open his and her camera, please do so. Okay. Okay, so just before the break, I talked about that I think the most exciting part and the most relevant part about this course is going to be to try to find these hidden patterns within the complex cell imaging data. And if I try to characterize, one second. Okay. And if we try to characterize this, what are the tasks which are we try to characterize two categories of tasks in bioimage analysis. I mean, this is my definition, my and Megan definition for a perspective that is going to come out, I don't know, very soon now. But basically, we are classifying bioimage analysis tasks to two categories. One we call a low level or signal driven, and this takes an image usually and creates output which are not interpretable. These are basically not directly interpretable. These are basically the standard image analysis tasks that you can think of, right? I mean, improving the image by applying different filters, tracking, segmentation, detection. All of these tasks are within the what we call the low-level tasks, and the high-level tasks are using the output of the low-level task and using them in order to ask a specific question. So we can take trajectories of a cell and make statistical inference and find that the cell, a group of cells are behaving one way, and when you put some drug on them, they behave differently, right? So these are the high-level tasks, and these are biology-driven because we want to ask, when we put this drug on the cell, what is going to change in terms of what we are measuring, right? So here's our two examples. One example, one general setting, and one example for a task of microtubules tip tracking, which I'm not going to explain now exactly what it is, but in principle, you have the standard image analysis task. So you register the image, you segment the cell, you detect the microtubule tip, you do tracking of the cell, you filter events based on some criteria, and eventually track the tips. And then based on that, we can do analysis of the trajectories of the cells and analysis of the track analysis, and then visualize and interpret the results. So in general, I mean, this course is more focused. We're going to show both, but we are more focused on the high level, on the biology-driven tasks, on the data-driven processing. So, and this is, of course, I mean, this course is built upon my personal interest, which is, I mean, this is the theme of my lab. The motivation always comes from fundamental questions in cell biology, and we have two types of outcomes. One is analytic tools to measure biological processes, usually measure something that was not measured before, and I'll give some examples later, and using that in order to make new biological insight. And usually this is about, it's not something that you can look in your eye, look visually and characterize the process, but you need the computation in order to actually make the characterization and interpret what's going on, and not just to put numbers in order to validate what your eyes see. Okay, so my motivation in this course is to bridge the gap between computer science and or data science and cell biology. I mean, I'm a computer scientist, and I'm trying to be as much of a biologist as I can. And I think, I mean, I think most of the I mean, I wouldn't say most, but a lot of the world now, especially the scientific world, is aware that the most exciting and challenging and meaningful problems are in the biological domain. I mean, you can see that in our department, it is recruiting quite a few of us now to actually, but it's not specific. I mean, you can see in our university and in other universities that people who work with biological systems are recruited to industrial engineering, to mechanical engineering, to electrical engineering, to computer science. I mean, every department has, because the most challenging and important problems are there. So the biologists have the nails, which are the exciting problems and systems, very complex systems that a wet lab have worked like 5 or 10 years to build up. And we as computational people have a very comprehensive and powerful toolbox of tools that we can use in order to solve and understand these complex data. But there is a huge gap between those two fields. And this gap is because of a lot of things. I mean, because the domain is complex, because the motivation of the fields is very different. And for example, computer scientists are a lot of times are driven by algorithmic elegance or by improving the state of the art, showing that my algorithm is better than your algorithm, you know. And so I was in a conference that was organized a year and a half ago by some donor at the Weizmann Institute that wanted to bring together exactly that, the computational scientists and the biologists, and they brought, and in terms of bioimage analysis, and they brought a bunch of people in the same room, and I was actually quite amused because the biologists were looking for service people to put numbers on what they see, right? To automate their processes. The computational people were interested in finding annotated data sets that they can compete on and try their cool algorithms on to improve. And there was, you know, they were not really talking to one another and understanding one another. And I think in order to make this bridge, we need to understand the different domains, understand the motivation, understand the terminology, and be able to communicate better. So I think this is the motivation of the course. And I'm coming from the computational side, and I'm trying to show you guys a little bit about why it's so exciting and why it's so important and interesting. And also, I think it's a huge opportunity now, and I'll tell you why a little bit later. So a lot of, I mean, usually the CS people who do this step and move into cell biology are doing a full shift and are going into biological departments and are building labs that do everything in one place, one shop, right? So the lab can do the experiments and can do the computational aspects. And there are fantastic scientists, much more established and successful than me, at least at this point, that are doing that, and great. But I think that the limiting factor here is that conceptually, as a computational scientist, as a computer scientist, we are trained to think about generalization, about reduction, how to take one problem and map it into another problem, right? And map a solution to another problem. And I think this is very powerful in the domain of biology, where there are a lot of systems that are very different from one another. And as a biologist, you cannot now be an expert in all of them and do all of the biology wet lab in your own lab, right? In one lab. But as a computer scientist, we can understand the general concept there and think about what is general and what is common for all these systems, and what computational tools could be used in order to solve problems in this domain and in that domain. So I think that computational people that do this transition a lot of time, just because you need to focus on a specific biological system, and then you limit yourself because this is what drives you, the biological systems that you're focusing at. And as a computer scientist, that is staying dry, that is not opening a wet lab, I think that we have the perk to think more generally and to think what problems can be solved with this and that tool. So sometimes it starts from a computational concept and what problems in biology can be solved by it. And also it can be when you see very different biological systems that where progress can be made with the same computational ideas, we can do that as well. So what is bioimage informatics? And in 2 weeks from now, we're going to get one of the best bioimage informatician or bioimage analyst in the world to give us a lecture about bioimage informatics. But I'm taking it in this Wikipedia definition. Bioimage informatics is a subfield of bioinformatics in computational biology. And the idea is the use of computational techniques to analyze bioimages at large scale and high throughput. High throughput, it means that the images come fast. I would say high content, so large amount of images. And the goal was to obtain useful information out of the complicated and heterogeneous image and related metadata. And if we look at one of the prominent reviews that came out, this is already 5 years ago, that says that the ultimate goal is to replace human labor as much as possible with computer calculations. So basically, this means that this talks about automation and completeness, the terms that we described in the previous hour about what computer vision and what computation brings into the table into the domain of cell biology. And what I didn't like here is the next sentence, so that biologists can focus fully on formulating high-level hypotheses and designing increasingly sophisticated experiments, while improving the objectivity and reproducibility of these experiments. So when things are said like this, I think that the focus is on we are tool builders. We're building a service. We're building a tool to a question that is defined by the biological partner, who is defining what is important and what is interesting, and we're a service provider, which is fine. And there is a whole field around that, which is I think which is doing a fantastic job, and we're going to hear about it. But I think that we as computational people, we have a unique perspective that can actually contribute to new ways to think about the biology. So I think that we can go a little bit further than that. So I look at bioimage informatics as going halfway. I mean, we want to work at the domain of biology. And what we do, so we want to work with biological image data, and we are building tools in order to aid in this. And I feel that this is like going halfway through this bridge, because if we go a little bit further, we take a little bit more domain understanding will allow us to actually ask questions and do a little more and be more interactive and productive in terms of making this type of collaborative science work. So I think that we can do more. And I think that taking ideas from domain, so me sitting in a computational department, sometimes, for example, we have people here working on ideas from cybersecurity. So immediately I'm hearing a seminar on cybersecurity, I'm thinking, oh, what biological questions can be solved by using these ideas from cybersecurity? Where can I apply these powerful and fantastic tools to the domain of cell biology? So this is one side, coming with a hammer and looking for a nail. And the other way around is, when you encounter an exciting biological problem, and we have the rich toolbox of a computer scientist, of a data scientist, what tools are useful to actually tackle and solve this problem? So, I mean, this is my, I don't know, maybe a little ambitious goal eventually to bridge computer. Who is not muted? Okay. Okay. So basically, this is my goal in this course generally to try to bridge this gap and show computer scientists that biology is super cool. And I think the tools here are bioimage informatics or tool building, tool development. This is one arm here. And the second arm is looking at it from a data science perspective and trying to actually find the patterns within the data to come with new biological insights. And if we do that, then biologists will have new and powerful tools, and we computer scientists will have interesting problems to solve, and everybody will be happy, and it's a better world, and blah, blah, blah. Okay, and I wrote about it an opinion, so if you want to, you can read. Yeah, I mean, I came here. I came here from a background in computer science. I mean, all my training is in pure computer science, in artificial intelligence, and I worked in industry in some as an algorithm engineer with images and with a high dimensional business intelligence domain, and came to a PhD in a computer vision lab. And really accidentally, I discovered cell biology and microscopy. I mean, I bumped into a biologist who had these images of what is called a wound healing assay or scratch assay. You'll see that later in the course. And these are very beautiful images of cells migrating in groups. And you see there are amazing patterns that are going in within this monolayer of cells, this group of cells that are doing crazy things. And when I asked the person there, okay, so what information are you extracting from this rich, amazing dynamic data? And they started mumbling something, which basically nothing or something very basic. And this made me turn into this field. I mean, my way was kind of, you know, I didn't know where I'm going, and it was kind of awkward. But then I went to a postdoc in a medical school in the US, and I came here, and I specifically picked. I decided going back to computational department because I'm trying to preach to the computational scientists and try to bring them to see the light. Okay. Okay, so course description. I'm going to introduce you to a broad set of biological questions. All of them, of course, are driven by the answers come from microscopy images, some live imaging and some static images, but it's all based on images. And we're going to show a set of computational tools in order to extract back biological knowledge from this data. So it's going to be very broad. We're not going deep into the biology of any specific problem. We're not going deep into the computational aspects of any specific problems. I do want you to get an intuition on both sides. So what is the biological questions? What is the intuition behind the computational tools that we're using in order to solve them? And how and what we can learn from that. And the tools are, okay, computer vision, machine learning, time series analysis, network algorithms, whatever we need in order to solve a problem. And I must say that other people might see the course in this type of course in different ways. I mean, I'm heavily biased to the things that are interest to me, but I think that I try to make a general representation of the field. If someone has, you know, something specific that you think is missing here, I mean, some of the biologists, or please let me know, and I'll consider including it or this year or next year. I mean, I'm really very flexible about it. I'm happy to change things as we go. Yeah, students from the department can come, of course. Students from other departments are also welcome. I got a request from a few students from the biological side of the university and biomedical engineering, and I'm happy. I want it to be an open course and an open discussion, and you're quiet so far. I hope that this will change with time. But yeah, I really want it to be to people from different disciplines talking to each other. Lectures are in English. I'm starting to feel less, I mean, in Hebrew it went much better in my first round of the course, but I think that I should try and stick with that and do it in English nevertheless. And I mean, and see, I mean, it's not, you know, I can stumble across words, or I'm not, I mean, I'm fluent when I present my own research. Now I present a very broad set of research, which is not necessarily my bread and butter. Not everything is, and it's fine. I mean, you know, I'm not ashamed of not knowing everything or not knowing to express myself perfectly in English. And I hope that you'll feel the same, okay? So please do not be shy. I mean, there is no reason to, and you're going to present a paper here in class, so you'll need to do it anyway. Don't be shy. It's a friendly environment. It's really, the purpose of it is to also to grow with people. I mean, you're going to work in whatever industry, academia, you're going to work in a high-tech company, and you'll need to talk to your peers or customers abroad, right? So we need to be able to communicate in English anyway. So please, to the people here that are not from Israel, most classes are, the vast majority of classes here are in Hebrew. I mean, we're not used to most of classes are in Hebrew. So it's also good for me to practice my English, because I'm feeling that it's getting worse as I am not using it enough. Yeah, I am going to expect some background in the math and programming, but I mean, but it's, I mean, yeah, I mean, you should know the basics of machine learning. I am going to talk about all the basics, but it will help you if you'll come with some prior, will really help you if you come with some prior knowledge about the basic concepts here. I am going to give mostly intuition. I'm not going to go into into formalities, and please feel comfortable also to stop me and ask questions about that. Didn't I do that? Yeah, no biological, I told it. Yeah, it's going to be challenging. I mean, the terminology, probably I already used a few words that are not clear to you. Please stop me and ask. I mean, sometimes I'm just not aware of that. There are no textbooks or resources. I mean, the course is based on research papers, most of them from recent years. I am going to, you're going to be able to see the list of papers. I am also going to give you some links to, if you want to fill up some knowledge gaps. I am going to give you some links on Python programming, on bioimage analysis, that you can, online courses and materials that you can learn from there the basics, or at least just skim over that. Great. So paper presentation in class is going to be 20%. This is going to be pretty challenging because I'm going to limit the time especially since the course is much more massive than I want in terms of a number of students. And also I want to go over a lot of material myself. I don't want it to be a seminar course. So we are going to have a very limited time frame for each of you to present a paper. The papers are hard. I mean, they're hard for computational scientists to go into this type of interdisciplinary papers and read them. They are more extensive. They are much longer usually than what you are used to. And part of the difficulty of taking these massive papers and bringing them to, I don't know, 10, 15 minute presentation is being able to squeeze the essence out of them. So being able to give a very broad background. So what we're talking about, again, we're not from the field, so we need some background to understand what is the context, what the researcher did and what they found, what's their contribution to the field, why this paper is special and interesting. I'm going to give you a list of papers that you can pick from. I'm going to curate a list of papers that you can pick from and present. They're going to be based on topics And the topics are we're going to take together a few topics per class that relate to one another, so we have some context. And they're going to relate, of course, to things that I teach in class before. Mostly these are going to be papers from the recent 2 or 3 years. So these are really new. Yeah. Questions about that?
Are you going to give them soon or later on during the course?
Yeah, I have a list, but I think a little bit later on because I think it's too early. I mean, you're just not, I mean, you don't have the background now to understand what's going on there. So let's wait with that a few weeks and then I'll put the list. If you really want it, I can share a list that and you know, an evolving list. I'm building it all the time, but it's pretty massive already. Final research project is going to be 80% of the course grade. It's going to be in groups. It's based on you. I prefer not groups of 3, but if you really feel that you have to be 3, then I'm not going to limit you by that. And you could take it as a one-person project. And there are a few flavors, and I'm going to go over that a little more detailed. I don't remember if I did it now in the slides or a little bit later toward the end of the lecture today. Basically, it's going to be, you can take one of different flavors. So one is going to be open data explorations. So there is public data sets that you can go into and try different techniques to ask different questions. And there are also data sets that we work in my lab on them, public data sets that are public or not public data sets that we're working in the lab to solve different problems on. And they are very rich. You can ask a lot of questions on them. And I'm going to describe it, and I'm going to describe what we are doing. And you can go to whatever directions that you'll be interested in. So this is one option. I call it open data exploration. The second option is collaboration. What I call collaboration is joining forces with actual biologists and trying to solve a real world. So a problem that they really need an assistant with solving. I don't have a list of a lot of these, right? I have maybe 1 or 2 what worked me so far. I can look for more. And it should be, and this is going to be, if you are going to do that, the experience is actually the communication with the biologist and actually solving a real problem. A lot of time it's going to be taking existing tools, understanding what is the computation, what is the quantification that is needed, and taking together the right tools in order to solve that and put numbers on visual data that they have. Yeah, so this is a second type of project. And the third type of project is a tool building validation comparison. And this is basically, for example, benchmarking, taking a task, a segmentation task, and now, I don't know, a tracking of cell tracking from bright field microscopy. So you're trying to track cells from this type of imaging modality, which is quite hard to image in bright field microscopy. You'll see that later. Cells and just benchmark, now collecting data sets with ground truth and benchmarking different algorithms to see who works better. I think that the most exciting is, exciting is the open data exploration, and I hope to see creative ideas come from you guys. But again, I mean, whatever you prefer personally. In principle, it will be, I mean, I'm going to give a very broad background on the data sets and questions that I think can be asked about it, but it's going to be very broad. You'll have to come up with the specific questions that you want to answer. We have a teaching assistant in this course. I just got the approval, I think yesterday or the day before, and it's Yeshaya, who is a student, with a graduate student in the lab, and he took this course last year, and he'll be able to give advice on initial advice on the project, so you can communicate with him on on presenting your ideas and starting, thinking together of what could be a good research question. And once you are ready, I think that sometimes in the middle of the course or toward I don't know, the last quarter of the course, we're going to hold a day, a few hours, which are not going to be in the time of the course. It's going to be a different time, right? That you guys are going to briefly present what your project is about and get feedback and what you did already, what you're planning to do, what is your idea, and get some feedback about that also from me. So these are the project. Yeah, and at the end of the project, you'll be evaluated also based on a written report, where in order to be so I can assess also each of you individually. I want each participant, if you're in groups and more than one, clearly articulate each one the contribution. Yeah, last year we had it worked okay. I think that we can improve it this year by doing this project presentation day, and by having a teaching assistant that is actually someone that you can discuss with, and I'll also be involved. So you can also approach me if you need help or guidance. Questions about the project?
Yes, about the data. Is it something that you think we can process on normal home PC, or we will need some special computation because of the size of the amount of the data?
Yeah, that's a great question. So, yes, the data sets that I'm organizing for you guys from that we work already in the lab, I'm organizing. It's not going to be the images. It's going to be information extracted from the images, right? So the low level is already going to be done for you guys. And this means that you can focus on the more data sciencey aspects, and yeah, that it's not, you don't need the huge computational resources for that.
Got it. Thanks.
And also, when you pick the project, pick it carefully. That if you don't want to, and again, it's not, even if we look at a huge screen with a lot of data that you can work with a lot of computational power on it. It's a project in a course. I want to see an idea and I want to see an implementation, an idea and trying to resolve it. You don't need to work on the whole data set. You can work on a subset of the data set and it would be completely fine in terms of, it's not a master's project or, yeah. Anything else? Lolit Bayesh.
Assaf, when do you think we should start?
Yeah, so I think I think it will take definitely after Passover, right? I need to give you some background in order for you to be able to think about these things. It's not, I cannot just throw out projects. You'll have no idea what to do with it. So I want you to have a little bit information about some background information about the field, about what can be done, what was done, and then you can start thinking about it. So realistically, it's going to happen in the second half of this course. And last year, the student asked me for more time to complete the project at the end of the semester, and I'm fine with that. I think I gave them until maybe I don't know, maybe 10 days or 2 weeks or something before I had to submit the grade, before my final deadline to submit your grades, which is also fine with me. It can be delayed.
Okay.
Hello?
Stuff? Are we not going to deal with any imaging processing in the course?
In terms of hands-on?
Yeah, a bit, or techniques.
Yeah, I'm going to talk a little bit about techniques. Yeah, I'm going to talk about techniques of. I'm going to show you the data is images, and I'm going to show you how the images are processed, and we are going to have next class. The next topic is deep learning in microscopy, right? So next topic, I'm going to show you what is the state of the art in using deep neural networks in microscopy images. So you are going to hear about it, but but we're not going to have any hands-on experience with that.
Okay. Thank you.
And the focus, I mean, and you know, it's not about fancy networks necessarily. It's more about what can be, how these networks can be applied in order to find something new. So, for example, you're going to see the simple idea of a kind of a generative network, right? A generative neural network, which is going to apply to one problem and then to another problem and then to a third problem. So computational is the same idea, but we can show how a simple computational solution can be used in order to solve a wide variety of problems from different domains.
Okay, thank you.
Yeah, I think I have in there. So we have the Moodle site, which is the official. I mean, if you are assigned to this course, this is the way that I can communicate with you directly. If you are external, I mean, if you are not taking this course and not from the university, then write, and you are interested in order to be informed regularly, then write me an email and I'll see how to include you in this as a free listener, it's called. And also there is a public website in my Wix website where I'm uploading the lecture notes, the slides that I'm showing you guys. I'll try to do it before every class. You know, the beginning of the semester, I did it before class, a while before. We'll see if it will continue this way. I'm pretty sure that at some point it's going to be like last minute preparing classes, and then it's going to be up just before class or after class or whatever. Yeah, so next week we're going to have a guest lecture by Natalie Elia, who is from the Life Sciences Department in BGU, and she's going to give a fantastic lecture, much better representing what I tried to describe at the beginning. I was also nervous because because it's my first time in English and etc. So Natalie is going to give a much better introduction than I gave to cell biology and microscopy, and how she sees this type of data science and cell imaging integrating with her world. She's a fantastic microscopist in our university. The week after, and it's going to take like, I don't know, 1.5 or 2 hours out of the time of the course. The following week we are going to have Genevieve from Pasteur from Paris, who is going to tell us about bioimage informatics. He's managing like one of the biggest and most successful bioimage analysis centers, and he's basically, they are providing service for everybody in their universities and other universities who generate the imaging data, and he's also developing tools. So whoever here is aware of TrackMate, for example, this is him, right? I mean, it's his code in there, and he's doing fantastic job in both aspects. So I'm pretty excited also about this talk. And I'm going to talk, the first topic is going to be deep learning in microscopy. So this is all very new, how deep learning is coming into the field of microscopy imaging and contributing in the last, maybe, I don't know, it started like, it came to microscopy in like 2017, the most earliest work. So we're really talking about a very short time frame where deep learning is in microscopy. So I'm going to give, I think, two and a half lectures about that. I'm going to talk about high content single cell phenotypic profiling, which is basically the idea that you can automate a microscopy system to image a lot of experiments of cells under different conditions. You can think about it in big drug companies. They have this, right? I mean, they try different drugs on the cells, and then they image them, and you have a lot of images, and you try to extract what treatments have brought the cells to a better condition, right? Or something like that. So We're going to talk about that, at least one class. I'm going to talk about how to represent the characteristic of cells, the shape of cells, the organization of the cells, and the organelles organization. Organelles is evronim, is the like the organs of the cell, right? The hands and the legs and etc. of the cell. So how it organizes within the cell and how data science can be applied there. I am hoping to talk about public data repositories and atlases of different atlases that are available out there. I'm going to talk about information processing in multicellular system, which is one of the big themes in my lab. So this is one of the topics that I'm very heavily biased towards. So you're going to suffer and hear about it. We're going to have another guest lecture from Tami Riklin-Raviv from electrical engineering at Ben Gurion. And she's more into the developer mode, more into the computer vision scientist who is building tools to measure things better. So you're going to hear stuff from her perspective and hear a little bit about her research. And then we're going to move into domains that involve not only imaging data, but also other types of data, omics type of data. And we're going to have Tal Shai, also from BGU, from the Life Sciences Department, who are going to give us a broad introduction on systems biology. And I'm going to show you some of the works that I'm very excited about in the field of system biology that I think that we should import to cell biology into microscopy. And I didn't have an opportunity yet to do it in my lab, but in the future I hope to. And we're going to have another guest lecture from Paul Wiltrix, again, another colleague from France. He actually did his postdoc in Israel at the Weizmann Institute, and he started his own lab just a year or two ago in Marseille. And he's going to talk about how to integrate those different domains, how to integrate images and microscopy. This is what his research is about, which is pretty exciting. And then we'll see if we have more time to other aspects, building an automated microscopy that makes decision on the fly, and using simulations and how to use simulations and integrate simulations and machine learning together. Image analysis in 3D, visualization techniques, maybe medical imaging. I don't know. Maybe I'm less. Students are usually, last time students really wanted the medical imaging, so I gave a class on that, and I brought also a guest lecture on that. For me, it's a little bit diverged from what I'm interested in, but maybe I'll just share papers, and then if you are interested in that, you can pick a paper on that topic. Okay. Yeah, so in terms of the project, I'm not going to teach image processing. I'm not going to teach computer vision. I'm not going to teach any specific technique. I'm not going to show you hands-on on anything. So in terms of the project, you will have to figure it out by yourself, which is another, I mean, it could be a big thing. Again, some of the projects, I mean, you can pick a project that does not involve images. It is already the post-processing of the images, and then you don't need to touch images. If you want to, you can learn about it, and I have a link at the end of my presentation with some links at some video tutorials and other tutorials that you can use in order to step into this world. Yeah, so I'm going to give a very brief background on what is an image and on machine learning, like very general and brief. But this is not all of the background. I mean, through this course, I'm going to give background almost in every class, right, about something. So it's not like I teach you I taught you the biology, and now we go and dive deep. I mean, it's going to be, hopefully, I mean, it's going to be like, each class is going to have some background that is specific to the class. Okay, any questions, comments, suggestions, concerns? Okay, so let's take a 10 minutes break and then go and then come back to the last hour of the course. And I'm here for a few more minutes if you want to ask me or talk or ask me something in Hebrew if you're shy. Hey, hey. Please join me. Okay. I'm asking again, if you can, please open your cameras. If you can't, then this time fine, but the next lessons, please, please, you can see the recording, it's fine. If you're here, please open your camera, okay? So I can know that I'm actually talking to human beings and not to black squares. It's fine, you can stay at home and watch it on your free time, but if you want to watch it live, then please be with us. Okay. Okay, so I want to go over that very, very quickly. If you have any specific questions, then please stop me. So, what is an image? And basically, what you see are slides that I take from my Intro to Computer Science course. So it's very, very basic. So, what is an image? An image is a matrix of elements which are called pixels. Each pixel has a value which implies the intensity levels or the gray level within the image. A two-dimensional image is encoded by a two-dimensional matrix, and on M a matrix gives you an M on M pixels image. If we are talking about videos, then we have these two-dimensional images over time. So we have a three-dimensional entity here, right? 2 dimensions of an image, and then temporal information, which are more frames in the image. If we're looking at 3D images, then it's actually similar. Just instead of time, we have the 3D, the third dimension, right? So we have a stack of 2D images stacking up, which is the third dimension. In microscopy, we have also multiple channels. So I told you that we take a fluorescent molecule and we tag our protein of interest with it, so we can see it. So in principle, if we tag molecule A and then molecule B, then we can have a 2-channel image, right? When watch one, we see the first, and in the second channel, we see the second. So we can have multiple channels that are actually a complementary information in terms of what is there in the image in microscopy. What is resolution? Resolution is the capacity to measure the smallest object with distinct boundaries. And the definition of a resolution depends on the physical size of a pixel. So if we have the same scene, as you can see here with the maps here, which is that you see here, and if a pixel if we have more pixels on the same scene, it means that each pixel is more sensitive, right? So we have a high resolution, we have more pixels on the same area, and it means that the physical pixel size of a pixel is smaller. So this is about the resolution, and you can see how resolution decays, how the information is lost about an object when a resolution decays. As you can see here at the bottom, since this R, if you look at the 100 by 100 pixel image, you can see the R very quickly, very, very, very clearly, sorry. But when you go down with the resolution, at some point it starts to be blurry, and at some point the whole information is about what is there is lost. So when we look at natural images, there are different ways to represent colors. The easiest one to explain is the RGB, red, green, and blue, which means that we have 3 channels, each measuring the amount of green, red, or blue in every pixel. In a microscopy, we can have multiple channels, but it's not anymore red, blue. I mean, you can see it in an image, in an image overlay of different colors on the same image, but the idea here is that each color represents a specific entity within the cell that is captured by a different fluorescent protein. Here is a grayscale image, right? So what you see here is a 8-bit per pixel. 8-bit, it means that we have 256 different gray levels, different values that can be encoded in each of these pixels. 0 is black and 255 is white. And on the left, you can see the image as we visualize it. And on the right, you can see the same image just encoded as the numbers, as the intensity values of the pixel. And basically, the computer needs to take this type of information, so a matrix of numbers, and to extract meaningful information from it, which is, if you think about it, I mean, when we see something with our eyes, then we do an interpretation immediately. But think about it, when you see something like that and a bunch of numbers and the computer needs to interpret that, it could be not trivial at all. Sorry, a bit depth is the number of bits per pixel. It encodes the amounts of information that can be included within this pixel. So if we have 1 bit, it means that we can have each pixel can be either black or white. And if we have 2 bits, it means that we can have values between 0 and 4. And in principle, the number of bits that we have define the number of colors, the number of gray levels that we can have in each pixel. A human observer can observe at most a few hundred shades of gray. And so for every reasonable application, I mean, from my experience, we don't need more than 8 bits per pixel. So 256 different values. It's sufficient for anything that we would like to do, especially if you think, even computationally, if you can understand more finer details in terms of the intensities, usually the amount of noise within your image is going to be higher than these numbers. So if I can take, I don't know, a 60-bit image, which means for each pixel I have 2 to the 16th different colors, the amount of noise in the image is going to be much higher than that resolution. So we don't need such a high resolution. We can use much smaller images in order to capture our data. Here is an example of image quantization. So on the left here, you can see a 24-bit RGB. So it's 8-bit for red, 8-bit per pixel for green, and 8 bits per pixel for blue. And you can see the amount of details that we expect to see in a normal image. But what I want to show you in the right, that even if we reduce here from 24-bit, right, to 2 to the 24 different colors, to 16 different colors, which is basically 2 to the 4 different colors, and we're still going to capture most of the information within this image. So we need to be aware of that. I mean, the spatial resolution is the same here. The resolution itself is not changing. What is changing is the bit depth, the number of bits that we have per pixel. So why is it hard for computer to interpret images? So, I mean, even if we look at this, what is this? Do you have an idea? What is imaged here? No, any xushim, מה זה?
Looks like a human
No
face,
idea.
or what?
Doesn't look like nothing.
My first year students immediately knew where it came from. I don't know how. In the intro course, of course.
It looks like a human face, I guess.
So this is the tongue from the famous image of Einstein. You know this picture that is with his tongue out? Anyway, you know, if we see the image, it's very clear, but now we see this piece from a picture out of context, which make it very hard for us to interpret that. And it's even more difficult if you look, if I show you this bunch of numbers, this matrix below, right? Without a visualization that captures the information within this matrix. I mean, it's really hard. And the first thing that you need to appreciate is that our eyes and brain are really good at interpreting images, and for a computer, it's much harder. So I'm going to go very quickly about the challenges in computer vision, and also this is a very old slide deck that I took from Lior Wolf from Tel Aviv University. And we start with this. Do you know this game? Anyone is aware of this game, I Spy? So the game is we say something and then you need to say where you see it, right? I mean, it starts simple, but then it becomes more and more complex. So I spy a mouse hole. Do you see a mouse hole in the image?
Yes.
Yes.
Okay, so here is the mouse hole, right? I mean, now this is very easy for us to see it. Why? Because it comes in context. I mean, we are looking for it in the right place, right? We know where to look for it and we know how it looks, right? So together, but if you think about the computer, the context is not clear at all, right? I mean, it's not obvious that the mouse hole will be in a very specific location. Can you see two butterflies?
On the floor.
Okay, so this is harder actually. This is actually harder because it comes out of context, right? And you're not expecting to see it for us, so it makes it harder. The second one is here, right? I mean, there is a kind of a butterfly. And yeah, again, the meaning of context. Now you have an empty green bowl, a bottle, which you can see here, of course. And this is immediate for us. But for a computer, it's the state. It was the state of the art to identify empty bottle. For example, I'm trying to, like I don't know, 15 to 20 years ago. Not so long. I mean, it was really hard to identify a green bottle, right? To identify a very clear object. And if we are talking about something more complex and less well distinguished, like a jar of a jar full of eyes, right? I mean. For us, it's very easy to see, but in order to train a computer to do that, to be able to identify such a complex object, in the last several years, it's possible, but it's relatively recent that we can do this type of interpretations. So I'm trying to show you some of the challenges between in computer vision, and if I want to talk about general challenges, and I'm going to show it in natural, but I picked ones that are also apparent in microscopy images. So you can see the same object from different point of view and different orientations, right? The cell can be in one direction, but can be also in another direction, and we should be able to identify that and deal with that, right? So this is one challenge. Another one is illumination. Is it the same person, or is it two different people? What do you think?
Looks different.
Looks same person.
So actually, I don't remember the answer, but it doesn't matter. I mean, because what I want to show here is how illumination, and we get that also in microscopy images, when we see illumination that is not necessarily uniform across the whole sample, brings pretty big challenges, especially if we want to look at the intensities and say something about that. Deformation. So here you see that we have an object that can change its appearance, right? It can change. It's not static in the same orientation and in the same. All the elements are in the same location in relation to one another, but it can change. And also in microscopy, our cell can deform. It's not, a cell is not an object that remains there all the time the same. It can change, it can extend protrusions, and it can change its morphology, can change what it does. So this is another challenge. Background information. I mean, we can see a lot of time the background is making us very hard it makes very, very difficult to identify what is important and interesting in relation to what is the background in our images. And also, I mean, everything here is a chair, right? But each chair is very different from one another. So how would we define a chair? I mean, when we look at the cell, I mean, also I can take cells from different systems. Each one of these cells is going to look completely different, and still each of them is a cell. So if you want to make a general framework that will be able to count cells, for example, it will be pretty challenging if you want it to work for very different cell systems. Local ambiguity. So here is a patch from an image, and the question, well, what is it, right? I mean, where does it come from? So when we don't have context, it's a problem. It can appear in a very different contexts in very different locations, and each time it could mean something that is completely different. So you can see here this patch that is matched to completely different images and completely different orientations. And context, when we look at something that happens, what happens around it allows us as human beings to understand the image better. And it's also true in some of the microscopy images, where it's not only a local region that is of our interest, but this local region is also affected by what's going on around. Okay, so this was my very quick introduction to images and image. Well, I didn't talk about any specific example of image processing, but challenges in images and computer vision. And now, again, like a 5-minute introduction to machine learning, which is also the backbone behind everything that we see today in computer vision. So traditionally, image processing techniques are now becoming, you can still use them and they're still effective, but they're becoming less and less common. And all the modern computer vision is all about machine learning. So how would we tell an orange from an apple? And the source here is actually a good resource. So if you do not have backgrounds, then I would recommend you look at that. And I have it in my resources a recommendation, which you'll see, which I have later in this presentation. So how do we tell apart oranges from apples? So basically, what we can do is well, say that if we have the two fruits in our hands, right? We can see how their texture and how much they weigh and their color, and I don't know, taste them and see, right? And then make a decision what is an orange and what is an apple. Everything that I say there is actually what is called feature extraction. So we extract properties that represent our object, and that allows us to make a decision whether we're dealing with an orange or an apple. More generally, supervised machine learning, and what you see here in the illustration is one of the traditional techniques, which is called the support vector machine, but there are a lot of other techniques. So supervised machine learning is we take data, in this case, fruit or images, we extract features, so we extract properties of whatever we're interested to learn about. And then we train a model that, given the features, it decides whether it's an apple or an orange. So basically it says, if the color is orange and the texture is this and that and the taste is this and that, then it's probably an orange. And if not, it's probably an apple. In order to do supervised machine learning, we need to have annotated data. So we need to have labels for each of the data. And basically what we do, we train the machine, we give it a pairs of an object or image or whatever we're interested in, and an annotation. We do the feature extraction, we train a model, and then we have a model that now when it gets a new fruit, it can tell us whether it's an orange or an apple. Of course, when we train a model, it can know only things that we trained it on. So if we now give it a banana, what the model is going to say is it's going to tell us whether it thinks that the banana is more similar to an apple or to an orange. Okay. Unsupervised machine learning is when we do not have labels. So we have data, and we do not have a label that says that these are apples and these are oranges, and we want to characterize. So basically, I mean, the most common application here is clustering. So we want to understand what different fruits or similarity groups we have within our data. So the idea here is to extract features and then to apply clustering techniques. We're going to see some of them, of course, both supervised and unsupervised techniques during this course.
Anomaly detection is not take a big part of our data to recognize an abnormal event or something like this.
The answer is no. I mean, anomaly detection is. I'm not even aware of techniques from anomaly detection that are applied to microscopy data. In my lab, we're trying to do that, right? I mean, this is exactly, I didn't give this example by chance, right? So I think this is one of the big opportunities, that the field is very rich, but it's very dull with the, I mean, there is a whole lot very powerful computational techniques which could be applied. Now, of course, when you talk about anomaly detection, you can apply supervised and unsupervised models in order to do that, right? I mean, so it's kind of implicitly. Did I answer your question?
Yeah. Okay.
Okay. Okay, what is deep learning? So deep learning is basically the integration of feature extraction and model training, right? So this is how it looks. I mean, we're going to talk about it a little. I'm not going to go into the basics, but I'm going to give a broader introduction in the next class that I'm going to teach. But very, very briefly, I mean, you give it inputs, it has outputs. In the middle, there is all this mess of neurons that are adjusting their activity and weights in order to optimize the output to fit the input. Okay, and what is going on here, or why? And this has revolutionized many fields, right? In applied computer science, specifically natural language processing and computer vision. I think that are the fields that have gained the most. I mean, it's really now all the modern techniques are based on deep learning in these fields, I think, or almost all. And the power of it stems from the ability to do both tasks at one time. Train a classifier in this case, or a discrete, yeah, train a classifier while extracting the features that will be optimal in order to train it. So if previously, if we take oranges and apples and we pick features that are not very informative or not optimal for the task, for example, I'll take the roundness of the object. So apples and oranges are usually kind of similar in the roundness. This is not very good informative information, but since we are extracting this information, we are defining what are the features, we're engineering the features, we might not do the optimal job in defining the best features in order to make discrimination. And the power of the network is that it does both. It extracts the best features in order to define the optimization task. Okay. Yeah, so yeah, I mean, I have here a list of current state of computer vision, and sadly I'm not longer an expert in that, so I'm not following really the state of the art or all the CVPR, ECCV, all the main conferences in the field any longer, because I'm trying to be more of a biologist than a computer vision expert. And but this is where I feel is the main contribution. We're focused in microscopy, and here is the big opportunity, because if you think about advances in microscopy, I mean, microscopy is advancing really fast. Molecular biology is advancing fast. Microscopy techniques are advancing fast. Automation. So now we can automate everything and generate like tons of data. But modern computer vision, I think now it's changing. Now in the last several years, there is really a lot of computational people who are realizing this opportunity and bringing computer vision into the domain of cell biology and building fantastic tools and very usable tools. I still think that there is a lot of opportunities there, especially at what I call the high level task. So the data science aspect of going beyond the tool building. And and one of the opportunities, and I'll give you an example in when I talk about I think I talked about it today. So using generative network to generate and take one image modality and generate another image modality is a very simple idea that came out in the last 3 years. It came out like 10 papers in the most prestigious journals that came out one after the other that basically do the same. In terms of computation, they did all the same thing, right? They trained the network to map from one domain to another domain. That's it. But they solved important problems with that, and this made a huge impact in the field. And I think one of the main things that is very exciting about it is that if you look at modern computer vision tasks, the state of the art is the human mind. I mean, what we can, how we can understand an image. So there is no machine now that can describe a scene, right, in text better than a human being, right? This is and if you want to segment something or track something or detect something, the benchmark is based on a human annotators that is doing that, or classify scenes or describe, do object detection, whatever. The human mind is like the gold standard. While in cell imaging data, it's not the case. We cannot capture the complexity and the richness of the information there, and computers can actually do better jobs than our eyes. Yeah, so I think I talked about it, so I'll skip it. I mean, my personal goal is discovery. And I think we'll skip this discussion to sometimes else. I want to really quickly show you some of the highlights, some highlights. I actually picked for today some highlights that I think most of them I'm not going to go over through this course because some reason anyway, but it can give you some idea of what's going on, right? I mean, so this is actually something that I'm going to show you, and this is using generative networks to improve the image quality, to enhance an image quality. So whoever knows microscopy, there is something that is called deconvolution, that you take an image and you artificially improve it computationally by using parameters from the image itself. What you see here is it's called image restoration. So improving an image, restoring an image, but without any specific model. So taking actual data and simulated data and training a model to automatically improve the image. And if the goal here you see an embryo of, I think, Drosophila, which is a fly, Zvuf perot in Hebrew. What you see here, and the goal is eventually we want to see the individual cells. What you see here are nuclei, the object here that you see. We want to see and quantify the individual nuclei and see how they work together. So in the original image, you cannot do it. The image is just too noisy for that. But after you use the system, this context-aware image restoration, you can do post-processing, which will allow you to do this type of analysis. Here is another example that I am going to show in this course. So one of the main challenges, holy grails, I would even say, is that we're not able to image a lot of molecules within the same living cell. And there are technical difficulties for that, which I'll get to in a later class in the course. But the fact is that if you want to see in the same living cell a lot of organelles or molecules, it's a problem. You cannot do it. And the idea here is that if we can take a bright field image, which is an image with very standard microscopy, you don't need to label specific molecules within it, and train a model. Each model will be specific for one prediction, for one molecule or one organelle. Then what we can do now is to take if it works, which is a big caveat here, a big question here. But if it works, what we can do now is take an image that nothing is labeled within it, and take all these models and generate fake images of all the organization within the cell. And then we can start to understand how the different components within the cells are organized in relation to one another, which you can just not do within an experiment, right? Which is pretty amazing, I think, if it will work. And here is an example of this technique, how it works. And I show it every time because I think it's amazing. what you see in the top is the data. So this is what we see. This is the bright field image. And I mean, you never saw a cell, so of course it's completely, you cannot understand anything here. But I can tell you that I mean, a trained microscopist can probably look at these bright field images and understand what's going on. But if you know, you use these 4 models and generate the different structures within the cell, you can see very clearly what's going on here. So I can see a cell dividing here. Here is the DNA that becomes from 1 DNA to 2, you know, 2, right? Coming from 1 to 2. Here is a membrane marker. So you see, you know, you see a cell when they make membrane marker. You see now here it's 2 cells, right? Here is nuclear envelope and mitochondria. You can see a cell dividing. The molecular organization of a cell changing in live imaging, where the live imaging was done without labeling anything within the cell. Now after we see that, we can see that, okay, here is cell number 1 and here is cell number 2. Okay, that's fine, but it's pretty. I think it's pretty amazing, this video. I hope you are as excited as me. And we're going. This is also something that we're going. I'm not going to. We're going to.
How is it divided? Was done here? How is it dividing between the different between different channels here? The models.
So the only things that so it was trained. So you can create a data set of the bright field image with DNA, okay, and a data set of the bright field images with cell membrane. So for each of them individually, you train a model that maps, that generates, takes as input a bright field image and maps it into a fluorescent image. So you made data sets, independent data sets for each one of these organelles. And then after you do that, you can take an image that nothing is labeled there and imagine, right, generate fake organization of the cell. Okay?
Impressive, yeah.
Okay, this is my work, so of course I'm going to talk about it. But again, it's trying to interpret. You use machine learning to interpret what's going on. What you see here is a melanoma cells from human patients. These are all melanoma stage 3, so they are actually from pretty aggressive melanoma. Stage 3, it means that they already metastasized to the lymph nodes, and they were taken from there. And the question is whether we can use this type of images, which is bright field images. The images that I showed you here, kind of, these are, it's a little bit different microscopy, but never mind the small details, which, as you see here, contain a lot of information regarding the molecular organization. And can we make predictions regarding what's going to happen to the patient? And the answer is yes, and I'm going to tell you about it. It's my work, so I'm going to spend on it in proportionate amount of time to explain things that I'm most excited about in the course. But it's not enough. So we defined the classifier and we showed that it can predict whether a cell is whether a patient is from a high potential, more aggressive or less aggressive tumor. But it's not enough because we want to interpret that. And the way that we approach it, and again, I'm going to take very briefly here, is do morphing within. So basically, we use again generative models. So neural networks will generate fake cell images, and what we do is we take a cell and we artificially shift it from a less aggressive to a more aggressive cell, based on what we learned from a lot of cells. And then suddenly we can see what are the characteristics, what are the cellular properties that are defining an aggressive cell versus a less aggressive cell. Again, this is going to be a full class. Other examples which I thought are cool, and I'm probably not going to get to in the course, citizen science in cell biology. So this is actually a cool paper that they used. They created a computer game that allowed the people to eventually people annotated, like, I don't remember how many hours, but tens of thousands of hours of work they got from people who played this game and annotated the biological imaging, right? I mean, it's not something that is easy for us. It's not that you show us a chair but they actually did a very nice job at that. And even some of the people there became like really good annotators, better than the experts in the field. And this allowed eventually to create a very big data set that could use for training different models for making different predictions on that. I am going to talk in this course about high content phenotyping or high throughput screening, which is basically you can take within one plate, and you can use a multi-well plate. Basically, each rectangle here, right, is what is called the well, be'erit, which is an independent, be'erit in Hebrew, which is an independent experiment. And in each of these wells, you can put different drugs, and then you can see how drugs affect your cells. And you can do that. You can collect a lot of information from that. And there is, it's a complete field, and all the big drug companies are using these techniques, and et cetera, et cetera. So we're going to talk about that. What you see here is an immune cell in 3D. This is actually my colleague and friend Megan Driscoll. So you see, these are the, what you see here is coming up is the microenvironment of a cell. So this is like a jungle of fibers, sievim, that the cell is in. And the crazy morphology of the cell, the crazy shape of the cell with all these arms that are going around is actually a real cell in 3D. That is taken with a very, very fancy microscope that allows you to get this type of images. This is not the image. This is a processed image, of course. The color map of the cell itself shows you the akmumiyut, the convexity. I think it's called convexity. No, there is a better word. Anyway, akmumiyut in Hebrew. I'll remember it later, probably. Yeah. So one of the cool things also, of course, is in this field is that the pictures are beautiful. I mean, they're stunning. And if you come to my office, you'll see a big picture of cells. That was the first thing that I got to my office. What you see here is event-driven microscopy. So what you see here is a development again of Drosophila, of Zuf Perot. And in this case, we want to, it's not us, it's a group from that is in a place that is called Genilia Farm, Genilia Research Campus now, they call themselves. And they want to image this animal developmental process live, right? The whole process live. And in order to do that, the microscope you need to adjust the microscope because things are changing, right? I mean, you need to focus it, you need to change parameters of imaging in real time. And what they presented here, I think, is a beautiful work where they automated the process. So based on the images that they captured, they made decisions in real time on how to change the parameters of the microscope itself to image these samples. And this is, again, work from my lab. What you see here is collective cell death. So when you see something green, it means that the cell dies. On the left, it's a fluorescent molecule that is within the nucleus of the cell. And when the cell dies in this type of death, the nucleus ruptures, מתבקע, הגרעין של התא מתבקע. And the green fluorescent protein, the green color comes out. And then you can see, and if you look at it, you can see that it's starting somewhere, but then it's propagating through the whole population. On the right, you can see simulation of this process. So we are using agent-based simulations to simulate this process, and then we use simulations to try to learn how can we explain what we observe in the real data based on different how we program our agent to learn something about the biology. Here is another example, just because it's so pretty. Here you don't see the green molecule, but you can see the death propagating. There are two populations here of cells, one at the bottom left and the other on the bottom right, and one on the upper left. And you can see that it takes all the cells at the bottom right to die before the upper left cells start to die. And it's pretty amazing. And here also, I mean, what data science tools we use here? I mean, I showed you before the simulations. I mean, there is image analysis in order to automate the process of quantifying this process and making predictions how a cell makes a decision to die. So we can use the machine learning models to actually, beside measuring the process and telling how fast the propagation, and whether it's more propagative or more some other form of cell death, etc. We can also try to make predictions whether how much the environment is affecting the decision of a cell to die, whether there are cells that are more resistant to that and to this treatment that cause this death, and they are more resilient for that, etc. Maybe I'll skip this. And let's see what I have for you. Okay, yeah, I talked about it briefly, about the research product types. I think that the best, I mean, you know, it's not going, you can decide whatever you want. I think that the most that you can get from in terms of this course is what I call here the data mining in public repositories or data from our lab, which is basically you'll get a complex data set that you can ask a lot of questions on and try to think like, what can you ask about it? But there are other types of projects that you can focus on. I have listed here a bunch of resources that you can go and look at data to start thinking what you can do. I mean, again, don't do it now. It's just too early. There is no, I mean, it's not going to be very effective at this point. But these are a bunch of data sets that I think each one of them could be useful for a project. You can see, yeah, all kinds of cool stuff. Even COVID-19, a screen that tries to take cells that are infected with COVID-19 and then does a screen and tries to bring the cells from a diseased state into a normal state, right? I mean, so you want that, you can take this data and play with it and save the world. Yeah, other data with live imaging. So these are static images, these are screening, these are general data sets. These are screening data sets. And we're going to have a class or two about screening. And these are live imaging data. The things that have underscore, yeah, I think underscore below it. So Bray et al. 2017, and here Goglia et al. And Bahav et al. These are data sets that we're already playing with in our lab, and we're going to have them organized for you also, so you can play with them and ask different questions. Resources on bioimage analysis, which is this file. So you have a link to this file, which you can have your different videos on. You see Python for microscopy, Python for bioimage analysis, and different courses. I really like the lectures from these ones. Yeah, I think they are fantastic. And yeah, Twitter threads about topics and others that are more general about using basic Python intro, basic machine learning 101, image processing in Python, very gentle introduction. I didn't go deep into each one of these links. I just looked for, I browsed through, and it seemed good to me. So if you find something that is not very helpful, please tell me so I exclude it from the list. If you find something that is better, please tell me so I can include it in the list. Okay, I think, so we have now 4 more minutes for questions, discussions. If you want, we can turn to Hebrew and you can ask me questions about the course or whatever. זהו, אין שאלות?
אני אצטרך אחר כך אולי רק לשאול אותך לגבי רישום, אבל זה...
טוב, רישום זה, כן.
אז הפרויקט הוא בעצם פרויקט כאילו ספציפי לדומיין, אבל גם כן דאטה פרוססינג, דברים רגילים של משין לרנינג כאילו, ודי יד חופשית להחליט כאילו מה אנחנו רוצים לעשות שם.
כן. כאילו מה, כאילו את שואלת כי זה מה, כי זה מטריד אותך, כי זה טוב לך, כי זה כאילו מה...
אני מנסה להבין איך זה...
כי את שואלת על זה כמה פעמים, אז אני רוצה להבין כאילו מאיפה זה, מה אני יכול לעזור כדי לתת את המידע.
כי אני רוצה להבין במה זה שונה מפרויקטים אחרים, בדברים אחרים. חוץ מזה שזה דומיין אחר עם טרמינולוגיה אחרת, שצריך כנראה להבין אותו. יש לו איזו ייחודיות כלשהי, לא יודעת, מנסה להבין את השוני.
לדומיין הזה?
כן. האם יש לו אלגוריתמים מסוימים שלו, או השימוש הוא די באלגוריתמים די גנריים שאנחנו לומדים גם בקורסים אחרים? האם יש איזה... מה מיוחד?
המיוחד בגדול זה הדומיין והחיבור הזה. אין... תראי, יש דברים שהגיעו ספציפית מתוך הדומיין הזה והגיעו לדומיינים הכלליים, נגיד יונט, בסדר? כאילו שזה רשת לסגמנטציה של תמונות. זה משהו שהיום משתמשים בו בהרבה גם דומיינים אחרים, והוא הגיע דווקא מהמיקרוסקופי ומהמדיקל אימג'ינג, שזה חריג. אבל בדרך כלל לא, אין כאן לא, בתחום הזה, החדשנות החישובית לא באה מהתחום הזה. הרעיון הוא לחבר, החדשנות באה מלהגדיר בעיה מעניינת ולפתור אותה בצורה, כאילו, ולפתור אותה בצורה נניח שלא הייתה עד עכשיו, כן? למדוד דברים, סתם, אני אתן דוגמה, נניח אנחנו לא מזמן פרסמנו מאמר של כלי חישובי שמודד תקשורת מכנית בין תאים בתלת ממד. אז יש לך תאים בתוך ג'ונגל כזה, ראית את התלת בתלת ממד הזה שנבנה הג'ונגל סביבו, אבל למיקרוסקופיה ברזולוציה הרבה יותר נמוכה מזה, לא רואים את זה, אלא רואים ממש תאים בתוך סביבה כזאת, ובעצם אנחנו בנינו את ה... רגע, קרן, את עדיין פה?
כן.
נעלמת לי פשוט במסך כנראה מישהו עזב. אז אנחנו בנינו כלי שבעצם משתמש בשיטות מ-image processing כדי לזהות את התאים, מסתכל על הסיבים האלה ומודד את הפלקטואציות בזמן של הסיבים במקומות מסוימים, ומתוך זה אנחנו מצליחים לזהות סיגנצ'ר, חתימה ספציפית של תאים שמתקשרים אחד עם השני, מכני, בסדר? החדשנות כאן, במקרה הזה אפילו אין machine learning, אין זה כאילו ממש לעשות טיילר כדי למדוד משהו שלא נמדד עד עכשיו בזה, בסדר? אז זה דוגמה אחת. דוגמה אחרת לחדשנות, יש לנו מערכת, עוד פעם, אני אתן דוגמה מהמעבדה שלי, מערכת של קבוצה של תאים מסתנכרן בה משהו לאיזה סיגנל חיצוני, בסדר? אז אנחנו בנינו איזושהי רפרזנטציה של רשת של איך מידע זורם בתוך הרשת הזה, בשיטות סטנדרטיות של time series analysis ו-information theory ובאמת, כאילו, הכי בסיסי של network analysis, ומתוך זה אנחנו הגענו למסקנה שתאים מחלקים ביניהם תפקידים בהעברת מידע, לכל תא יש תפקיד אחר, יש להם זיכרון, הם זוכרים את התפקידים שלהם, אנחנו מבינים איך המידע זורם מרמה של התא הלוקלי לרמה של כל המערכת, אז כאן החדשנות היא היכולת לבוא עם, כאילו קודם זה הכלי שמודד משהו שאי אפשר למדוד קודם, ובדוגמה הזאת זה בעצם לקבל אינסייט ביולוגי חדש. החוכמה היא הרבה פעמים בזה, כאילו, או להתקל בדאטה מעניין ולחשוב מה יכול לפתור אותו, או לחשוב על עוד פעם, נגיד סייבר, כן? שזה זה, אני ראיתי כאן כמה הרצאות שנתנו במחלקה על סייבר, ואז הסתובב לי בראש, איך אפשר להשתמש בזה בתאים? כאילו, זה יהיה מדליק להגיד, הנה, אני עושה סייבר סקיוריטי על התמונות מיקרוסקופיה שלי, עכשיו רק צריך לחפש בעיה שהיא מספיק חשובה ומשמעותית, ושזה באמת תהיה תרומה אמיתית כדי לפתור אותה. אז זה, אני חושב, מבחינת טכניקות, את יודעת, את יכולה להשתמש בטכניקות יותר חדשניות, פחות חדשניות, זה החלטה שלך מבחינת הפרויקט.
אוקיי.
אולי עוד דבר אחד שכאילו ייחודי יחסית לקורסים אחרים, אני יודע, אולי שזה תמונות, כאילו אין כל כך קורסים במחלקה שעוסקים בתמונות, זה דבר אחד, גם התמונות של מיקרוסקופיה הן מאוד שונות מתמונות טבעיות מבחינת הסטרקצ'ר שלהם, אבל זה לא, עוד פעם, אני לא חושב שזו מוטיבציה לקחת את הקורס הזה או לא, כאילו...
אוקיי.
אסף, אני אשמח אם בהמשך אולי תפרסם או תעבור על זה באחד השיעורים על דוגמה לפרויקט אפשרי, כדי שבעיקר נדע מה הסקופ שאנחנו נדרשים אליו. ואיזה גודל של נושא לבחור או משהו כזה.
סבבה, בסדר, אני אתן דוגמה עקרונית, עוד פעם, זה פרויקט של קורס, כאילו אין כאן, זה מחקרי, אז open-ended, אז אין כאן, אני לא צריך שתגיעו לתוצר מסוים, כן? אני לא צריך שתגיעו לתוצאה סופית או זה. אני צריך מה הרעיון, מה ההיגיון מאחוריו, באיזה כלים השתמשתם כדי לפתור אותו, איזה דאטה יש לכם, מה, כאילו, אני רוצה לראות, החיבור הזה, זה מה שמעניין אותי. פחות ש, כן? אז יש, ניסיתם לעשות זה ויש משהו שעובד קצת יותר טוב או זה לא כל כך. או הנה, עשינו סקרינינג ויש דאטה סט שתופס, כן, שאי אפשר לעבוד עליו על המחשב הלוקלי שלך, אבל את יכולה, אבל את לא צריכה את זה בשביל הפרויקט. תקחי כמה פלייטים, כמה ניסויים בודדים מתוך הדאטה סט העצום הזה, ותשחקי איתו, תעשי את הרעיונות עליו. אז מה אם את לא תופסת את כל זה? אנחנו לא עושה פה אצלי מאסטר, את במקרה כן, אבל זה לא, זה כולה פרויקט, כן? זה כולה קורס.
סבבה.
אבל אני כן אומר לכם שזה כן דורש יותר השקעה, כאילו זה כן, אתם תצטרכו לחשוב על זה, אתם תצטרכו לקרוא, אתם תצטרכו, זה לוקח זמן ומאמץ שאני חושב יותר מפרויקט בקורסים אחרים במחלקה, נראה לי. זה ההרגשה שלי. זה גם הפידבק שקיבלתי מהסטודנטים שעשו שנה שעברה. אני אתן לכם דוגמה גם, בסדר? עוד שאלות?